Good afternoon. I'm pleased to see this hall full, I would say almost as usual, but maybe you have something to do with it. And this is the week of the Heller Lecture Series in Computational Neuroscience. The Heller Lecture Series is a gift that we have for a long time from a couple that really cared about us when we started. It was about 10 years after we started. In 2002, Morgan and Michael Heller from London decided to donate the annual sum of £15,000 to establish at ICNC at the time a high-profile series of lectures entitled the Heller Lecture Series in Computational Neuroscience. The primary aim of this unique lecture series is to acquaint the interested public with most recent excitements of brain research. Sometimes we do it more popular, sometimes less, but the main thing is that each year we invite 4 world-renowned scientists to visit the Hebrew University for about a week, where they can deliver talks, interact with the students and faculty of our center today, ELSC. And I believe, Ed, you could see that this was the purpose of the visit, and I hope you enjoyed it like we did. And before we go to the talk, I want to say again how much our gratitude to Morgan and Michael Heller. Thank you very much, dear couple. They continue to do it since 2002 till today, and I hope they will continue in the future. And once we said our thanks, it's time to present the speaker of today, Ed Callaway, who comes to us from San Diego. It's time to move this picture because you will see it if you go and visit. And I will say just few words about today's speaker. So the work in 8 laboratories is aimed at understanding how neural circuits give rise to visual perception and behavior. His lab is developing molecular and genetic tools, methods to facilitate the elucidation and manipulations of neural circuits and apply imaging and physiological tools to study the circuit activity and dynamics during visual processing. Am I right? More or less? Good. Okay. In the previous talk, Ed talked or focused more on the methodologies that he developed, and that's the very interesting aspect of his lab, which is daring to develop technologies continuously, ongoing, and do good science with the technologies that they develop. So I hope that today, I believe, you will tell us more about how these different tools that we talked about in Tuesday are used in your lab. And I thank you again very much for sharing your science, your ideas, your methodologies, and your interesting findings with us. Thank you very much. One more thing, I'm sorry. So this is the tradition here from 2002. We have this certificate. The Heller Lecture Series in Computational Neuroscience awards this certificate of recognition and appreciation to Ed Callaway for his contribution to academic inquiry and exchange from the Edmond and Lily Safra Center for Brain Science, signed by myself and by Israel Nelken, the head of the teaching program. Good luck, and thank you very much.
So this works? Everybody can hear me? Can you hear me in the back? No. Okay, great. Well, thank you, Ilan.
Okay.
Yeah, I think so. They say they can hear me? Tell me if you ever can't. I'll talk loud. So thank you, Ilan. It's been great to visit here and visit the labs and see all the great science that's going on here, and all of the big plans for the future with new buildings and exciting things like that. And I'll be having dinner later with the students, so I'm looking forward to that as well. So, as Elon mentioned in his introduction, my talk on Tuesday focused on methods that we've been developing, as well as methods that have been developed by other labs, focusing on molecular, genetic, viral kinds of technologies. And this talk is going to be different from that. Toward the end part of the middle, I will say there will be a part that will be redundant with Tuesday's talk, where I'll talk again a little bit about the rabies virus-based tracing technologies that we've developed, but in a much shorter version. This talk instead is more of a sort of 20-year history of what we've been doing in the lab, and it's meant to sort of take you on a journey from what we could do, what our limitations were 20 years ago, 15 years ago, the kinds of experiments we had to do to try to unravel finer scale kinds of circuits, microcircuits in the cortex. And it will initially focus on experiments that we did in the monkey visual system. And I'm going to sort of focus in on just one sort of particular question there that we tried to answer, just as a sort of vignette or an example of that should illustrate how tedious things were and how difficult it was to get to one particular small question that I'll just focus on there. So that's more of a sort of example of what things were like back then, but also we learned some things back then as well. And then at the same time we were doing those experiments, we were also doing experiments in brain slices, which clearly revealed to us that the specificity of connections in the cortex was much more precise than we had anticipated prior to that, or we maybe hoped things would be simpler. And this led us to realize that we really have to have better tools to target specific cell types and individual neurons within cortical circuits or anywhere in the brain. And so we at other labs started developing various kinds of techniques. And so I'm going to tell you at that point a bit about this rabies-based technology that we've developed. But at the same time, is that targeting cell types in monkeys is pretty difficult. We still can't do that very well. I sort of touched on that a bit in the talk on Tuesday. But targeting cell types using genetic technology is much more straightforward to do in mice than in monkeys. So we've been switching over to the mouse visual system. And so at the end, I'll talk to you about some of the foundational work that we've been doing to better understand the organization of the mouse visual system and contrast that with the monkey visual system. But there are some things that are similar and some that are different, but the hope is that there are some things that are similar, at least in terms of how different components of cortical circuits function, that we can find commonality and learn something about people by studying mice. So we start out with a bit of introduction about where we were in our knowledge of the primate visual system roughly 20 years or so ago, which was where we started off in my lab, and this first story that I'm going to tell you about. So this is a well-known diagram from Dan Kellerman and David Van Essen, where each of these squares that's colored represents a different visual cortical area in the monkey, except for these squares down here, which represent the retina. Retina connects to the lateral geniculate nucleus, and then from there to primary visual cortex. But then all of these other squares are different visual areas. And at that time, people could use standard anterograde and retrograde tracers and show which areas are connected to each other. And people could stick electrodes in the cortex and record visual responses and see, well, the kinds of visual responses you see in one area are different than the others. You could make a lesion and show that area MT is very important in motion perception, for example. So you could do all of the kinds of things that we would hope to do to try to unravel how circuitry relates to perception at a coarse level of resolution. You could identify what components are connected together. You could correlate those different structures with different functional attributes, different kinds of visual responses, and you could do lesions to show causal relationships. But what was missing here is that you might see a transformation happen, that visual responses in one area are different than the visual responses in the area that it connects to. So something changed, but you don't know how. Were there neurons with diverse properties here that projected out to different places and provided different kinds of input, and that's why these areas are different? Or did they already receive the same kind of inputs and have some different kind of architecture to the circuitry that extracted out different kinds of information? So those kind of questions you really can't get at when you have this kind of level of resolution. And what you have to do is get to microcircuits. So that's what my lab started out trying to do in the monkey visual system, and we made some progress in that. And I'm going to talk about those kind of experiments that we did. And in those experiments that I'm going to focus on, we focused on the primary visual cortex, which is here in this diagram. This is from a textbook. It shows some things that are true and some things that are not true. Let's focus first on the things that are accurate here. So first of all, there are parallel pathways that originate from different types of retinal ganglion cells and carry different kinds of visual information through different layers that remain segregated in the lateral geniculate nucleus of the thalamus. So these magno and parvocellular strings continue to project in parallel to different layers in the primary visual cortex. So there's this parvocellular pathway, which originates from midget retinal ganglion cells, and it has nice small receptive fields, high spatial resolution, carries color opponent information into the cortex. It's a relatively slow system. This magnocellular system has better temporal resolution, more coarse visual information, no color opponent. Those two streams come in parallel here to these different layers, the parvocellular stream to 4C beta and the magno to 4C alpha. What's illustrated here that's less true is the idea that these two different pathways, the magno and parvocellular streams, give rise to different output streams. And the dorsal visual cortical areas, the ventral visual cortical areas. It's certainly true that these are somewhat independent pathways, and there are different kinds of visual information represented here. But what's not true is that the parvocellular stream exclusively gives rise to this pathway and the magnocellular to this pathway. There's actually quite a bit of mixing of these two pathways within the primary visual cortex that give rise to new output pathways. But this was an attractive idea at the time, that the magnocellular pathway gives rise to these areas, because there's not a lot of color processing up here and these motion areas. It makes sense that a faster system might be responsible for deriving motion information, and a higher resolution stream for more information about shape and color. But we did a whole lot of experiments looking at local circuitry in the primary visual cortex of monkeys, where, and I'll be talking about some of those kind of experiments in more detail, but we did a number of experiments that showed that there's mixing of information from the magno and parvocellular streams. Magnocellular information coming in layer 4C alpha here, parvocellular to 4C beta, and it mixes together and gives rise to many different outputs to different cortical areas. And here are just some of the publications from that time that showed us that there are interactions between these different pathways. But one thing that was a little bit more difficult to get at is a sort of short story that I'm going to focus on in more detail as an example of the kind of experiments that we did and sort of how tedious they were and what it took to get an answer to a particular small question. So we wanted to know, in particular, a little more about how this motion processing area MT, whether it got a contribution from the parvocellular visual pathway. So there's lots of evidence that it got input from the magnocellular pathway, but it was more difficult to get at this kind of question. And to answer that question, whether there is a parvocellular contribution to area MT, we're going to focus a lot on this layer in the primary visual cortex, layer 4B. So again, in this diagram, we have the retinal ganglion cells that give rise to the magnocellular and parvocellular pathways. The magnocellular layers of LGN project to 4C alpha, and the parvocellular to layer 4C beta. And then layer 4B is the primary source of direct outputs from B1 to MT, as well as indirect outputs that will eventually get to MT through these other visual areas, B2 and B3. So if we want to know whether there is a parvocellular contribution to area MT, layer 4B is a good place to focus, and you might want to know whether layer 4B neurons get input from 4C beta neurons because they're getting this parvocellular information. So you might get at that kind of question, do layer 4B neurons like these here, or these excitatory neurons that project to MT, get input from layer 4C alpha and 4C beta by doing reconstructions of axons of cells that are located in these different layers. So these are neurons that we intracellularly filled with dye and brain slices, and they have morphologies that you see here. These are axons projecting up here. The dendrites are much more local. And you can see these neurons in layer 4C beta send axons that go up through layer 4B without branching, whereas the cells in 4C alpha have lots of branches in layer 4B. And it's in fact information like this that was already present from Golgi's studies before we started working on this that led to the idea that the parvocellular pathway doesn't contribute to area MT or to those dorsal visual areas, because if these cells don't have axons in layer 4B, how could they be making connections to these cells that project out to those dorsal visual areas? But if you look closely, you can see that there's actually overlap of dendrites and axons here. These pyramidal cells, like this one, have lots of dendrites that could overlap and get this input. And even spiny stellate cells like these that are also excitatory neurons projecting to MT have dendrites that overlap with these axons.
So
Yeah.
This is the monkey or monkey?
Sorry, this is all monkey at this part that are talking about. I'm not going to say, So that's really important. All of these pathways, we're going to contrast later in the talk. The visual system of the mouse is organized in some ways similarly, in some ways differently. And that's something we'll touch on. But yeah, thank you for pointing out. This is all monkey visual system. You wouldn't have these separate layers in 4C alpha, or a lot of this architecture is actually pretty unique to primates. Okay, so we'd like to know then, functionally, are there connections from these cells in 4C beta and or 4C alpha onto layer 4B neurons? So to get at that question, we actually did functional experiments where we reported intracellularly from neurons in brain slices and then stimulated neurons in other layers to see if there are functional connections. And to do this, we used a method that I developed along with Larry Katz when I was a postdoc in his lab, where we bathe the brain slice in caged glutamate and then flash UV light, small spots of it, that uncages glutamate locally. And then some cells will fire action potentials in response to that. And it turns out if you do this right, the only cells that fire action potentials are very close to the site of glutamate uncation. So if we stimulate, say, at this location here in layer 4C beta, we know that only layer 4C beta neurons fire action potentials. And if we record here in a whole cell voltage clamp recording, we see these excitatory inward currents that are evoked following stimulation there. We know that some layer 4C beta neurons must in fact make excitatory connections to this stuff. And so we can stimulate many locations and make a map of the sources of functional excitatory input to this one neuron. And so we can see here that this cell actually gets pretty strong input from 4C beta as well as 4C alpha. And it turned out that the pyramidal neurons like these, generally 17 out of 19 of them, got strong input from layer 4C beta. About half as strong as the input from 4C alpha. But the spiny stellate neurons like this one never got input from layer 4C beta, just 4C alpha. So that starts to get to an answer to our question, is there a parvocellular contribution to area MT? Well, there could be, because some of the neurons, these pyramidal neurons at least, got input from layer 4C beta. But this then pushes the question a little farther. You have to ask, well, then if the pyramidal neurons get input from 4C beta, but the spiny stellate don't, you need to know what types of layer 4B neurons project to area MT. Is it only the pyramidal cells, or only spiny stellate cells, or both? And there had actually been a publication in 1989 where Ship and Zecchi had injected horseradish peroxidase into MT and looked at the morphology of neurons in layer 4B of V1 that were projecting to MT, and this is what they saw. And you can see that you can try to make some guesses about whether some of these might be spiny stellate or pyramidal, but it's pretty difficult to tell. So they actually got it right. They thought that more of these were spiny stellate than pyramidal, but we weren't so sure based on this. So we used a method that we started using much later in the lab, where we use a rabies virus that's been genetically modified, and I'll talk more about this later in the talk. But it's had a gene deleted from its genome that's required for the virus to be able to spread from initially infected neurons to other neurons, the glycoprotein gene, and that's been replaced with green fluorescent protein. So with this virus, if you inject it somewhere in the brain, it's taken up at the axon terminals, as you see here in this diagram, and the viral particles spread back to the cell body. And then this virus very efficiently expresses lots of green fluorescent protein in the cell and completely fills all the dendrites and axons, so you can reconstruct the morphology of cells that project to the location where the virus was injected. So what we can do then is make the injection of this virus into areas like MT or V3 or V2, and then wait about 5 days, and then perfuse the animal, look at the morphology of cells in V1, they're projecting to these different visual areas. So this is a photograph of one of those cells that's labeled in V1 after an injection into MT. The cell was filled with green fluorescent protein. The reason it looks black here is we just stained it with an antibody for green fluorescent protein, and the reaction product is black. And this brown background stain is cytochrome oxidase stain that helps us to see the layers 4B is in between these two cytochrome oxidase dense bands. 4C here to start. And if we zoom in on the cell, it actually turns out we can look across multiple sections and see all the dendrites of the cell. This turns out to be a spiny stellate cell. It doesn't have an apical dendrite that extends up above layer 4A here, even though its cell body shape looks somewhat pyramidal. So that's a spiny stellate cell that projects to MT, but we also saw some pyramidal cells that project to MT, like this one, very clear apical dendrite extending up. We also compared the cells in V1 projecting to MT to those that projected to V2, and we also saw pyramidal and spiny stellate cells projecting to V2. But the main point is that what we saw is that most of the cells projecting to MT are spiny stellate cells, 75%, whereas most of the cells projecting to V2 are pyramidal. So does this get us the answer to our question, or a kind of obscure question? Is there a parvocellular contribution to MT? Well, turns out there's both spiny stellate and pyramidal cells, and yeah, I told you pyramidal cells got input from 4C data, but I told you that 2 out of 19 didn't. So maybe the few pyramidal cells that project to MT are the ones that don't get 4C beta input. So we really still don't quite have clear answer to our question. We really need to know, do the pyramidal cells that project to MT receive parvocellular input? And I've also been kind of ignoring another possible route of information from V1 to MT. There are also layer 6 cells in V1 that project to MT, so we have to worry about those as well. So a different way to address this question is to use an intact rabies virus. This one doesn't express green fluorescent protein. It's just a wild-type CDF strain of rabies virus. And this is a virus that, like the other one, will infect axon terminals, but a normal wild-type rabies virus is competent to create new viral particles in the cell that is initially infected, and then it spreads transsynaptically and spreads only in the retrograde direction. And if we use a survival time of 3 days, we get only disynaptic label. So in 3 days, what you get is that the axon terminals, the cells at the injection site, take up the virus, it replicates in the cell, spreads across one more synaptic step, and labels the direct disynaptic inputs to these cells. So in this case, say we inject in one of these higher visual areas, the layer 4B neurons, say, that are projected to MT will take up the virus, and now it'll go one more synaptic step and potentially label cells in 4C alpha and or 4C beta that would connect to those cells. So now we're going to see, are the cells that provide input to these in 4C alpha or 4C beta, or both. So here are some results from an injection in area MT of this intact rabies virus. Each of these panels is just progressively blowing up more and more on the labeled cells. These cells that are black are stained with an antibody for the nucleocapsid of the rabies virus. So these are cells that have rabies virus in them. And you say, look here, you see lots of cells in layer 4B. These would include cells that were projecting directly to the injection site at MT. But in addition, here's the border between 4B and 4C alpha. You see cells down here in layer 4C alpha. These cells don't project out of V1. They must have been labeled because they connect to the cells in layer 4B that are projecting to V1. So you can see lots of cells in layer 4C alpha, but none in layer 4C beta. And we can quantify this. There are a tiny number of cells actually in 4C beta, and these turn out to be just at the border between 4C alpha and 4C beta. So you can see across all of the cortical layers, most of the label is in layer 4C alpha, very little in 4C beta. If we just zoom in on just the layer 4C label, you can see that most of the label in 4C alpha. So this would seem to give you an answer to our question. You'd say, well, it looks like there's not a parvocellular contribution, at least disynaptically, to area MT, because there was no label in 4C beta. So these layer 4B neurons that are labeled here, projected to MT, are not getting input from 4C beta. But it turns out that when we look at the lateral geniculate nucleus of these animals, we get a surprise. Remember I told you that the parvocellular layers of the LGN are what project to layer 4C beta. We see no label at all in layer 4C beta. So if we look at the lateral geniculate nucleus of that very same animal, we in fact see lots of neurons labeled in the parvocellular layers of the LGN. So how did that happen? I mean, these are cells are projecting to layer 4C beta, and there's no label in layer 4C beta. So they must be projecting somewhere else onto neurons that make direct projections to MT, because this is disynaptic label. So we see label in both magno and parvocellular layers, and we did other experiments in antibody staining. There is a population of cells in the LGN that projects directly to MT. This is not them. So it turns out that the very likely and only plausible source for that is these neurons that I ignored earlier that are in layer 6 and project directly to area MT. So it turns out that these neurons must be receiving both magno and parvocellular input and projecting to MT. So in a way, I've probably created a story that was a little bit simplified at the start to make a point, but when we look at all of this together, the bigger picture is that we shouldn't be thinking of a sort of monolithic pathway through the LGN to V1 out to MT, but there are many different pathways to MT that probably each carry different kinds of visual information. There's a pathway through these pyramidal cells to get both magno and parvocellular input and go to places like V2, which can eventually provide input to MT. There's these layer 6 cells that get magno and parvocellular input and project to MT. And there's this other pathway through the spiny stellate cells, which is the clean magnocellular pathway with little or no parvocellular contribution. It's the most direct route to MT. So if one wants to better understand the system, what we would need to do next, and we're gradually working on getting better methods so we can do it, is we would like to know what are the differences in visual responses of these different neurons that contribute to these different pathways. And we'd like to be able to selectively inactivate these different components of the system and see how each of them contributes to MT. But that's a little beyond us now for the monkey. We're just now getting methods where we can retrogradely label cells in V1 that project to these different visual areas and then do 2-photon calcium imaging of those cells. So we'll be able to do that soon, but we're not there yet, and that's the kind of thing we do in the future. This text just shows what I've already said. So that's sort of this short vignette of the kinds of experiments we did 10 years ago to try to get at these kind of details about local connectivity in the monkey visual cortex and how tedious and difficult it was.
Is there a good reason to stop the analysis at the level of disynaptic connections?
No, not at all, right? Because I think, so the question is there a good reason to stop the analysis at the level of disynaptic connections? In those particular experiments, it really was to more get at the question of whether these directly projecting cells connect there. But in fact, in those experiments that are published, we did longer survival times. And you can very quickly see in just another day or two labeling in layer 4C beta. It becomes much more difficult to interpret those results and to know how it got there. But that clearly shows that, in fact, 4C beta does through some pathway projected. So it might be that the V2 neurons that project to MT get input from the layer 4B pyramidal cell to get input from 4C beta. So you only have to really go 1 or 2 synapses more to that. But because this is something that sort of come up with the rabies virus later, with this attack rabies virus, the rate of spread, it spreads further and further into the system as you wait longer, but there's a confound in that the rate of spread is related not only to how many synaptic steps you spread, but also the strength of the connections. So a weaker connection might take longer to label, even though it's more direct than something strong that's less direct. So as you'll see later, we've developed a way to make the rabies virus stop so that you can just unambiguously identify direct inputs. So at the same time we were doing these experiments in monkeys, you can imagine making brain slices from monkeys, doing laser scanning photostimulation. We didn't have slices from monkeys every day. So we did at the same time a lot of experiments from rat slices. And we looked at questions like the one illustrated here. This is something that I pointed out also in my talk on Tuesday, where we, again, use the photostimulation method to map inputs to single neurons. And if you record from a single neuron, you must be recording from a cell of a particular type. And what we could show is that different types of neurons receive functional inputs from different layers. And this told us that connections in the cortex, here excitatory connections, are cell type specific. That is, a cell of, say, this type is much more likely to receive functional connections from layer 5 than this type. So if a layer 5 neuron is sending axons to layer 2-3, it doesn't indiscriminately connect to any cell according to the proportions of dendrites in that cell type, but it can selectively connect to some cells and avoid others. And that's something that hadn't been appreciated at this time in 2000. Most models of cortical circuitry assume that you could just fill cells with dye, look at the dendrites and axons and their overlap, and you could make accurate circuit diagrams that way. And it turns out you can't. And this turns out to be more of a rule than an exception wherever you look in the brain. And this panel over here just shows that this principle is true not only for excitatory connections, but also inhibitory connections are cell type specific. So inhibitory neurons in layer 5 are more likely to connect to this layer 2-3 inhibitory cell type than this layer 2-3 inhibitory cell type. So connections are cell type specific. And in addition, on Tuesday I talked in some detail about data that led to this schematic, where we showed that it was already known that neighboring neurons in layer 2-3 of the cortex are only connected to each other a small percentage of the time. And it turns out when they're connected to each other, they share common input from layer 4 and layer 2-3, and they form these sort of relatively independent subnetworks within the cortex. So these are two kinds of levels of resolution that we know then matter. We know that cell types matter, and we know that there's even this kind of fine-scale specificity where neighboring neurons of the same cell type can be connected differently. And this really led us to question whether we're ever going to be able to figure out how the cortex is working with the kinds of tools that we had available at that time. So if you want to link connectivity to function, you want to know how circuits work. As I pointed out in the very first slides, it used to be we had a level of resolution of maybe areas where we could say, here's a wiring diagram of the connections between areas. We can correlate connectivity with functions. We can manipulate components at that level of areas. But here what we need to do is we need to worry, if you want to understand how microcircuits are working, you need to worry about cell types and little pieces of a circuit that are all intertwined and meshed together. So that's what really led us to think about using molecular genetic tools, because with molecular tools you can target cell types. And now, as you know, there are, say, many different kinds of mouse lines that express a pre-recombination of one cell type and not the other cell types. And we can use a full range of modern tools for linking circuitry to function. So we can do that and link things to cell types, link function to cell types in separate animals. But something that you can't do in separate animals is to deal with this level of specificity, where a unique neuron might be connected up in a unique way. If we want to know how that functions, we're probably going to have to link connectivity to function for that particular neuron directly. Look at its connectivity and function for that same neuron in the same animal. And I made that point in my talk on Tuesday as well, but as I talk more about the rabies-based tools that we've developed, I wanted to point out at the end that the cells remain alive for at least 14 days or so, maybe longer. And so you can label circuits and then link them to function. So I'm going to try to go through this part somewhat quickly because it's redundant with what I talked about on Tuesday. This is just the slide to make that same point. You can link connectivity to function by doing different experiments in different animals when you're worried about cell types. But linking connectivity to function for single neurons is much more difficult. But because rabies doesn't kill cells, you can label a circuit and then interrogate its function. So, how many of you were here for the Tuesday talk? Okay, more than half. So I'll go through this pretty quickly. So earlier in the talk, I showed you how we could use a glycoprotein-deleted rabies virus to just label neurons and project it to a particular area, have them kill or die, and this virus didn't spread to other neurons. But I also showed you data with an intact rabies virus that could spread transsynaptically, selectively in the retrograde direction. And we've already talked about the fact that that kind of a virus is difficult to interpret the results from because there are confounds between the strength of connections and the number of synaptic steps. So what you'd really like to do is to have a way to get rabies virus to only infect particular cells of interest, or maybe even one cell, and then take advantage of its ability to do what people that are making EM reconstructions have the hardest time doing, and that's to trace axons. So people trying to make EM reconstructions of circuitry in the brain have a really hard time following the axons more than tens of microns even to try to make circuit reconstructions with that method. But rabies virus can do that beautifully for you. If it infects one cell and then spreads to other cells, it traces the axon exactly for you. It spreads right up the axon and labels the cell body. So we'd like to get rabies to infect one cell or a population of cells of a particular type, and then spread across only a single synaptic step, label the direct inputs, and then stop. So I'm going to illustrate how our system works in the context of labeling the input to a single neuron. And our method is an inherently genetic method, so we start off by introducing a few genes into a single neuron. And there are three different plasmids here that we're going to electroporate in this example. One is just a fluorescent protein, so we can find this cell. We're going to call it a starter cell because it's where our rabies infection is going to start. So this is fluorescent protein is going to mark this cell. The second thing is a gene called TVA. This is a receptor for an avian virus. And this is going to allow us to selectively infect this cell with rabies virus, as you'll see in a minute. And the third thing is Rabies Glycoprotein. This is the gene that's been deleted from the rabies genome. It's a gene that's required for the virus to be able to spread. And if we infect the cell with a virus that has that gene deleted, this is going to allow that virus to spread from that neuron. Okay? So we electroporate the DNA. We wait 2 or 3 days. The gene products are expressed. We've got now fluorescent protein in the cell to mark it. This TVA is on the surface of the cell. There's rabies glycoprotein expressed. And now, if we come along and we inject this virus, which is a rabies virus that's been genetically modified and also pseudotyped. So the glycoprotein gene is deleted from the genome, the gene that's required for the virus to be able to spread, and it's been pseudotyped so that on the outer surface of the viral particle, rather than having rabies glycoprotein, the normal envelope protein from this virus, it has a different envelope protein called NV. There are no receptors for NV in the mammalian brain. If we inject this into a normal brain, it can't infect anything. Okay? But the receptor for that virus is TVA. So since this cell is expressing TVA, this NV pseudotype rabies virus will selectively infect that cell and it won't infect anything else. Okay? So that's how we're going to get our initial targeting to the cell of interest. Okay? This virus has M-cherry in its genome, so once it infects this cell, the cell expresses the M-cherry and turns red. Also, the virus can replicate in that cell, and it can take advantage of the rabies glycoprotein that we expressed in that cell and and through transcomplementation, make rabies virus particles that have rabies glycoprotein on them, infect axon terminals, spread back, and label the directly presynaptic cells of M-cherry. And then there's no rabies glycoprotein in these cells, and as I've said, without glycoprotein, this virus can't spread. So it's just mono-synaptically restricted, and you label the direct inputs to the cell and nothing else. And this is just one picture from one example of this, where it just makes a nice pretty picture, but there are cells labeled all over the brain that in areas that we know project to the primary visual cortex. This is a layer 2 pyramidal cell with the starter cell, in this case the mouse visual cortex, and then these are our inputs to that cell. So you can label quite a few inputs. When this paper was published, we were typically labeling 50 or so inputs. As I mentioned on Tuesday, Botan Ros's lab has improved this in various ways, and he's getting up to 500 or 600 inputs per cell, which might be close to all of them. So it's soon we're probably going to need to do some electron microscopy and sampling and quantify what proportion of the inputs are getting labeled, because nobody really knows exactly how many cells should connect to one neuron. So this is one way to use this method. The other is to label these direct inputs to cells of a particular type. We would use that, many people have, using mouse lines that express free recombination of particular cell types. And you could, now what you want to do is get the cells that have free to express the same genes that we electroporated in the previous example, the TDA and the Rabies Glycoprotein and the marker gene. And most people who do this use a helper virus, an adeno-associated virus that would inject, would indiscriminately infect all the cells here, but only the pre-expressing cells would then express the genes carried by the helper virus, the TDA and the glycoprotein. And you just do the same thing as before, infect that population of cells with the N-base serotype rabies, and label the direct inputs to the cells. And I'm not going to go through all these controls, but just say that this works. There are lots of published papers using this. This is an old slide now. Everything isn't always exactly perfect, right? So there are different kinds of trade-offs between, there can be a little bit of leaked TDA expression in non-free expressing cells that confounds your ability to interpret local connectivity, but it doesn't affect long-distance connectivity. So the kinds of helper viruses and versions of TDA that you might use for long-distance connections would be different than if you want to look at local connections. And so there's sort of little idiosyncrasies like that those of us working with these systems have figured out over the years. And so if you're thinking about doing these kinds of experiments, there are different versions of helper viruses and different versions of TDA and things and other things that you might design somewhat differently. So it's a good idea to not just dive right into it, but to read some of the literature or send me an email and get some advice before moving forward too quickly. So the other thing I wanted to point out is that you can use the rabies virus for functional studies to directly link connectivity to function. You can insert whatever genes you want into the rabies genome. Here are just some examples of fluorescent proteins. We've made plasmids that make the cloning easy. These are all deposited at Addgene and freely available. And we've made a bunch of different versions of rabies virus expressing different genes that you might imagine would be useful, like genes where you might be able to express a genetically encoded activity indicator, where you might like to control activity, optogenetics or pharmacogenetics, where you might like to control gene expression in those cells. So you can then do the kinds of experiments that you might imagine to link connectivity to function. And this is just one example where you'll see in a little bit that we can use intrinsic signal imaging to map visual areas in the mouse. And here we just injected a rabies virus expressing GCAMP into one of those areas to label cells in V1 projected there. Here's a Z-stack from 2-photon in vivo imaging. You can see these cells that were labeled both with BS-RED and GCAMP from the rabies genome image all the way down to layer 5. And then here in a plane looking from the surface, we can present visual stimuli to the mouse and show that these cells are, say, direction tunes. So using drifting gradient tubing in different directions, you can see the tuning properties of the cell. So it's possible then to link connectivity to function in this way. And in fact, as I talked a little bit more about on Tuesday, Botan Ros's lab has been using this to directly correlate the connectivity inputs of single neurons to the functional properties of the neurons that converge on that one cell in the mouse visual cortex. So we're getting close to the end in this marathon sprint, and moving to then our experiments in the mouse visual system. So the mouse visual system for now is probably the best place where we can really get at cell type specificity of connections and function because of all the mouse lines that exist. But we haven't given up on monkeys. We're still working hard on making better tools to target cell types in monkeys, but we're just not there yet. And it probably won't be too long before there are some Cree-driver lines, at least for marmoset monkeys. There are some knockout monkeys that people have made. Might soon be a GCAP expressing monkey line. But we'll never be able to do the kinds of experiments as quickly or as easily in monkeys as we can in mice. So we've been doing a lot to explore the mouse visual system and better understand it. And there are a couple of landmark papers that really led people to take the mouse visual system a little more seriously in the last 5 years or so. And one of them was a study from Andreas Burkhalter's lab that suggested that the mouse visual system was composed of many different extrastriate visual areas in addition to the primary visual cortex. And his experiments that suggested this were based mostly on anatomy. And I'll show you an example of some of those experiments in the next slide. There were also some functional studies that suggested that there weren't quite as many visual areas. So there was a little bit of uncertainty there. But it turns out that Andreas was right, as I'll show you in a little bit from experiments that we did with intrinsic imaging. So here are the kind of experiments that Andreas' lab did. He injected multiple colors of anterograde tracers into V1. And because of the topography of connections between visual areas, you could see this kind of red, green, yellow pattern showing up in several fields surrounding this. And each of these fields then is presumably a different topographically mapped visual area. And you can also see these sort of reversals, mirror image representation along the vertical meridian here that are illustrated in this slide. So we wanted to know first whether these were in fact functional visual areas, functionally mapped, and if they're there, how repeatable are they from animal to animal? And then once we know that they're there, we can use intrinsic imaging, if we can see them, as a guide to do more detailed studies of the visual responses of neurons in all of these different areas and try to identify how they might be different from each other. I should point out, though, at this point that one thing that's clearly different here already in the mouse and the monkey is that each of these fields is receiving input directly from V1. There's a whole bunch of fields, if these are all different visual areas, getting direct input, unlike the monkey visual system, which is much more hierarchical, where there are only a few areas that get very strong inputs, and then another level up and up. So this seems to be a less hierarchical system in the mouse. So we used intrinsic signal optical imaging to get at to make maps of visual space in the mouse visual cortex. And if you have visual stimuli that move periodically back and forth or up and down, you can use the phase differences of the responses to make an estimate of the location in visual space that activates each of these locations. You can make contour maps like these, altitude and azimuth. I mean, you can start to see things like a border here that's the vertical meridian, and maybe a patch here and a patch here that are different areas. But it's pretty difficult by eye to make clear borders between visual areas. And it turns out that when we came up with a more objective way of doing this, the kinds of maps that we saw and the things that we saw were much more clear, and we even discovered a couple of areas that we didn't know were there. So here what we did is to use this field sign analysis that we adapted from experiments that were done in the human visual system based on functional magnetic resonance. This is from Marty Serino. Was one of the first to look at this, where you simply take a particular pixel on the cortex location and look at a vector moving across space vertically or horizontally, then you take the shortest distance between the two, and it's either going to be counterclockwise or clockwise. That is a non-mirror or a mirror image representation of visual space. And here that field sign value is plotted according to this axis, so you end up with positive and negative field signs, then you start to see very clear borders. We have some algorithms that we use that then draw borders objectively between these, and then you can very clearly see all of these multiple visual areas that show up. And you see most of the visual areas that Andreas predicted. We also see a couple more, ALL and MM, that we didn't see, probably because these don't get strong direct input from V1. And then we don't see his field A, which clearly does get input from V1, but seems not to be as retinotopically mapped. So we now know all these visual areas are there, and we next want to ask what are the functional properties of the neurons in these different visual areas, so what kinds of new features might emerge. So to do this, we use 2-photon imaging. We can first use intrinsic imaging. All the intrinsic imaging is done through the skull to identify the different areas. So we can use then the blood vessels as landmarks, do a craniotomy, inject organ-grade calcium indicator into one or more of these areas, and then zoom in, and then look at functional responses to drifting gratings. So here's an example of a cell in one of these areas, LM. You can see we use different spatial frequencies in this cell, like the particular spatial frequency, different directions of motion. This cell like these two different opposing directions, so it's an orientation selective cell. And we can make plots of orientation selectivity, direction selectivity, spatial frequency, and temporal frequency tuning. So Jim and Marina reported from thousands of cells in 1, 2, 3, 4, 5, 6, 7 areas, including V1. And the first thing you'll see that's quite different than a monkey is that only about half the cells in V1 of a mouse are visually responsive. And in these extrastriate areas, there are many cells that are not visually responsive. The proportions are somewhat lower. So these are probably requiring some interaction between different kinds of sensory modalities or something else to be able to activate them. I should say it's not an artifact of the method. We've done and published 2-photon imaging in monkeys, and 98% of the cells that are visually responsive with the same methods.
Visually responsive to moving gratings in particular?
Yeah, this is just to the drifting gratings that we're using here. So you might imagine maybe there's some other kind of more specialized visual stimulus that's required, but there's some evidence from other labs that suggests that things like pairing with auditory stimulation or things like that might. This is, and I should say this was in anesthetized animals, but other labs have now done experiments in awake animals, and these proportions, at least for V1, don't change. So it's not because the animals are anesthetized either. So in this sort of first pass analysis of how these different visual areas might differ, we are just looking at these features: temporal frequency, spatial frequency tuning, direction, and orientation. And so here, if we just look at temporal frequency tuning, we can see that V1 in gray and this area PM in red respond much better to slower moving stimuli. All these other extrastriate areas like more fast moving stimuli, so the speed of modulation of the gratings. You can also see differences in spatial frequency tuning. V1 is the highest resolution as you might expect from a larger visual area, and the extrastriate area is somewhat smaller. But you can see that they tend to sort of be a bit binaural. Some areas here that like high spatial frequencies and some with low spatial frequencies. You can find also differences in the proportion of cells in each area that are orientation selective or direction selective. So I point out here these areas AL, RL, and AM that are more direction selective than the other visual areas. And so based on this, you might, well, the first thing you can say is that each visual area is unique in a signature kind of way. There's overlap in the distributions of responses to all of these stimuli. But if you sampled from enough cells and knew the distributions in each area, you would be able to predict what area those cells came from. So each area is in a sense unique, but there's a lot of overlap in the responses of cells. This is a plot of preferred temporal frequency against preferred spatial frequency, but with additional parameters like direction selectivity to separate it out more. One thing you see here is that unlike in the monkey system where the magno and parvocellular pathways, I told you that in the monkey, the magnocellular pathway is low resolution but fast. So you might expect that's low spatial frequency, high temporal frequency. And you sort of have that relationship here, but you clearly don't have it for all these extrastriate visual areas. There are some areas that have relatively high spatial frequency tuning and high temporal frequency tuning. And this area here that's kind of like what you might expect for a ventral stream or parvocellular-like system, it's over here in a dorsal rotation. So, yes, OSI is orientation properties. Yeah. I'm going to come back to this in a couple of slides, but yeah, because yes, OSI is orientation property. Yeah, DSI is direction. I don't need to go through that. I've said all that. So, so it's interesting to try to make some analogies or look to what extent things might be organized similarly or different in mice and monkeys. Mice, like monkeys, have a primary visual cortex. Virtually all of the information from the LGN goes through primary visual cortex to go to other areas. I've already mentioned that they're different in that at least all of the areas illustrated here, we have a couple other areas not illustrated, get direct input from V1, whereas in a monkey it's more hierarchical. But what about the idea of a dorsal or where kind of pathway and this ventral what kind of pathway? They're based on the anatomy and connectivity experiments that have been done by Andreas Burkhalter looking at connections in the mouse. These areas out here that are colored in pink from this review from Chris Neal are connected in the way you would expect for dorsal areas, and these more like ventral areas. But things that don't fit functionally are that, say, this area PM is a higher resolution area, even though it's anatomically in the dorsal stream. So if we were to try to, say, make a connection between mice and monkeys in terms of how spatial frequency tuning and temporal frequency tuning, the coarseness of visual receptive fields relate to dorsal and ventral, that wouldn't fit so well. But if instead we think about it in a different way, as dorsal stream being more responsible for action and a ventral stream for perception, that probably fits better. There have been a lot of experiments reporting from rats and also now some in mice that implicate these posterior parietal cortical areas in spatial navigation. There are less experiments done with implicating these than, say, maybe object recognition or things like that, but that's what you might expect here. So in fact, another way of looking at dorsal and ventral streams in primates that was pushed a lot some time ago by Mel Goodale, who did studies in humans, was this idea of action versus perception based on lesions in dorsal and ventral areas in humans. And so if we try to make that kind of analogy, that probably fits better and is probably a more universal way of thinking about this. We shouldn't necessarily expect the exact kinds of visual information that are represented in the areas to be the same, but what the visual information is used for might be similar, action versus perception. And also because in the mouse, most of these cells are not strictly visually responsive, they're probably interacting a lot more with the other sensory modalities. And in fact, we've retrogradely labeled inputs to these different areas. And you'll see direct motor cortex inputs to these. Auditory cortex connects directly to V1 in the mouse. So they're much more interconnected, and probably V1 is a much more associative area in the mouse already. I don't need to go through all of this again. I think I'll just end there and want to leave you with this slide that mentions all the people that did the work that I talked about today. Thanks.
About the 3-day transaction plan for dynamic results with the Rabies, how much is that time dependent on the length of the connection?
Yeah, so the question is, so with the intact rabies virus, and we trace connections, we actually, a lot of the experiments that led to understanding that were done by Peter Strick's lab, so I give him credit for that. We relied a lot on his experiments and did some control experiments of our own. But the question is how much of the timing is related to the length of the axon as opposed to the number of synaptic steps. And the amount of time that it takes to go down the axon is relatively short compared to the time that it takes for the virus, once it gets into a cell, to complete its life cycle. So the first thing that it does when it gets into a new cell is that it expresses the genes in its own genome. So it carries with it its own polymerase, it's a negative strand RNA virus, and it translates its own genome and makes all the proteins that are required for another viral particle. And then after those have accumulated, then it makes the negative strand genome that will get packaged and go to the next cell. And that whole process takes, on the order of 24 hours, and the transport takes on the order of, 5 hours, 10 hours, whatever, depending on the length. So the length has a relatively small effect. But as you go across more and more steps, that is going to create a more and more of a concept.
So, but multi-modality that you were alluding to in mouse V1, is that a global, is that unique to visual cortex? I have two questions on that. Is that unique to visual cortex in mouse, and is there any anatomical explanation for the inputs into V1?
Yes.
Just what's going on?
Yeah, so the question is this multimodality of V1 that I've alluded to. I should say, that's I'm guessing, right? So the evidence that that's the case is not super strong. I can base that on anatomy that we've done, and for sure there's a big difference. I mean, if you do retrograde labeling for V1 in mouse, you see input from secondary motor cortex and the prefrontal cortex. Yang Dan had a really nice paper recently where she showed direct functional influences of the prefrontal cortex on V1 directly, and did optogenetic activation of the cells in the frontal cortex that were projecting there, and showed how that modified V1 responses. The question is whether that's something that's unique to the visual system in a mouse, and I couldn't tell you. I don't know. I don't know if experiments have been done that would really look at that. There's been a tendency in general with anatomical experiments that people will look for what they're interested in looking for and don't reconstruct across the whole brain. I think now with some of the newer methods, the ability to have slide scanners that scan the whole brain, we're looking at whole brain in all of our experiments. I don't know that that's always been done in some of the old literature. But I would imagine
that you described with the two photon imaging, has that been replicated in other primary modalities?
Yeah, so people have done two-photon imaging in other sensory areas, and I actually don't know what the proportion of cells are that are say, whisker/responsive. I think people do worry a bit more there about whether they're giving the right kind of a stimulus, especially in barrel cortex, because it's sort of it's a both sensory and motor kind of combined area, and it might be that sort of the animal actively whisking might be required. And so there are probably people here who know more about that than I do, but there are debates about whether you have to have active whisking to activate it or passive would be as good.
Okay, now it's my turn then. So partly you answered the question I had during the talk about the number of neurons or the percentage of neurons that responded, and it's relatively small, especially in the non-primary, and that has to do maybe with the modulations of context, namely maybe top-down. Did anyone do experiments trying to increase or decrease the top-down modulations of the different areas in the mice, especially in the mice? In monkeys there are some that I know, but...
Yeah, so there's one experiment and this is the one I just alluded to, that Yang Dan did, where she activated the prefrontal cortex, and she showed that activates VIP cells, which inhibits somatostatin cells, which leads to disinhibition of pyramidal cells. So that is
it goes in this direction.
So that is
increase the number of neurons.
Yeah. So that's a facilitatory effect. That's one particular pathway that she looked at. In experiments that we've done in my lab that I didn't talk about today, we used rabies virus to trace inputs to different inhibitory cell types. VIP cells, somatostatin cells, parvocellular cells, and we see that invariably for all cortical connections, which in this case would have all been feedback connections, so we only looked at primary sensory areas, V1 and S1, the input to the VIP cells was much stronger than to any other cell type, which is consistent with the idea that this might be sort of a universal kind of influence of these kind of top-down kind of connections, maybe bottom-up too, because we also saw that for the thalamic input. The thalamic input was stronger onto, and that's true whether it's a secondary thalamic nucleus or a primary thalamic nucleus, stronger onto the VIP cell.
Yeah, I do have a comment about all these approaches. The comment is And if you want to see top-down approaches, you don't look just after the stimulus but before it. And most sensory scientists don't do this. Okay, so I think it's something that is missing in the, call it methodology or the strategy, but I do think that the top-down approach would be seen better as the context rather than the modulation of the response itself. I mean, it would respond the response itself, but if you look before, then you know how to separate them.
Yeah, and that sort of fits with this sort of, if prefrontal cortexes may be involved in working memory, sort of there's some cueing or something like that within.
Joe.
But you mentioned towards the end that some of these visual areas are implicated in spatial connections and spatial representations.
Well, in spatial navigation, so people recording in these cells that will respond when the rat makes a left turn but not a right.
Okay. My question, do you know how many sinuses go from, say, V1 up to areas like the hippocampus or spinal cortex?
It probably is, but I don't know off the top of my head. I know, actually,
Different in mice and monkeys.
Yes, for sure.
Monkeys, we know the number of stages.
Yeah, I don't know for sure. We probably even have the data where I could tell you that, but I don't know the data off the top of my head. You know, the extrastriate areas are getting direct input from it.
This day when the novel goes to investors, Yes? Okay. I have one more last comment. Unless there is another question, I want to make one last comment. It has to do with the division between action and perception. And I want to ask you if you don't think that we always have to look at the perception as an active process, namely there is no action and perception separately, and the direction that you suggested here is actually going to separate them even more. So we actually don't want to separate them today. We think that action is part of the perception. What do you comment about that?
No, I would agree completely. I mean, perception drives action influences perception, and all of these diagrams that separate all these things are just cartoons that are, probably as much wrong as they are right. Where, I sort of, tore apart the part of it that says that Magno and Parvo are independently contributing to dorsal and ventral streams, which is wrong. But also, I mean, that other part of that diagram that showed them being independent pathways is wrong because there are direct connections between each of those areas. And there are observations, like Tom Albright, my colleague at the Salk Institute, has done experiments where he showed that area MT is a motion processing area. But he showed that if you train a monkey to associate a particular direction of motion with a particular shape, neurons in MT will become selected for that particular shape. So, there's something that's carrying shape information that's connected to this supposed motion area, and there's interactions between them, and there's probably a lot of plasticity, and you're going to adjust the connections and function of your brain to match what your needs are.
Okay. Thank you very much.